So today we're going to continue our lesson on genetics, and we're actually going to start talking about how we can predict the traits of our offspring. So we already know, we learned this last time, how to identify the genotype and phenotype of our parents. So now what we're going to do is we're going to use that information on our parents to start predicting, well, what will the traits of our offspring be? What traits can we or will we pass on to our offspring? And today we're going to do that through something called a cross, so specifically a monohybrid cross, which just means that we're going to predict only one trait of our children. Eventually we'll learn how we can predict multiple traits getting passed on from generation to generation at the same time. But today we're just going to focus on one single trait and how that can or cannot or may not be passed on to our children. So before we start that, we need to remember a little bit about meiosis, which we learned about just a few weeks ago. And so remember that for every gene, you have two alleles because you got one from your parent, one from your mom, and one from your dad. When we go ahead and make our gametes, our sex cells, our sperm and eggs, remember that we split up our chromosomes and we split them up evenly. So what's going to happen is that when we go ahead and make our gametes, our sperm and eggs, they're going to have just one of the alleles that our parents has. Or just one of those parent alleles. So what happens is if your two alleles, let's say we're using the letter B here to represent our alleles, we have one dominant, one recessive. When we go to make our sperm and eggs, and remember we make them in fours, half of them are going to get the dominant allele, and half of them are going to get the recessive allele. And you're going to see when we write out our Punnett squares, because we always know it's going to split up 50-50, 2 in one and 2 in the other. We're only going to write them once to represent 50%. So we don't have to write big B, little b. You'll see this in a second. It'll make sense because it's always going to be a 50-50 split. 50% of the gametes will have the allele from one chromosome, and 50% will have the allele from the other chromosome. Or let's just say if this was my dad, 50% would get the chromosome from his grandfather, 50% would get the chromosome from his grandmother. And there's different rules to how those can mix, but that's the general rule. And what's important to note is sometimes they might be the same allele. So for example, if we had little b, well, if we were going to split that up evenly, well, then half of my sperm or eggs would have little b, and the other half would have little b. Now, what's important is when we go ahead and do our crosses, we're still going to write both little b's. And this is just to stay consistent throughout. So let's talk about how we actually predict our offsprings using this information. We're going to use something called a Punnett Square. And you've probably seen these before. You might have used them in middle school, or you might even use them in math class. And Punnett Squares are just a tool that we can use to distribute things evenly and predict the probability of something, specifically the probability that an offspring will have a given trait. So what's important to note about these Punnett squares is we're going to build different ones of different sizes depending on how many traits we're looking at. For a monohybrid cross, we're going to have a 2x2 square. So we have 4 boxes in here. Each box is going to represent 25% of our children. Or, I should say it this way, it's going to represent a 25% chance that our child will look this way. If you add up the 4 boxes, 20, 25, I get 50, another 50, 100. The box always adds up to be 100% of all your children. So let's go ahead and use this to solve a genetics problem that you might see. And so the first step is always just going to be to identify the genotypes of the parents. This is similar to what you did yesterday. So here I'm giving you a little information about the trait that we're using. The trait for height is represented with a T. Tall is dominant, so that's going to be our capital T. And short is recessive, so that's going to be lowercase t. It says a man is homozygous dominant for height, and he is crossed, that means mated, has children with a woman who is homozygous recessive. So the first step is always to identify the genotypes of these two parents. So the man is homozygous dominant. Remember, homozygous means the same, and dominant means capital T. So two of the same capital T's, he's going to be capital T, capital T. We're going to go ahead and write that down. Now let's figure out what the mother is. So the woman is homozygous recessive. So again, homozygous means the same, and recessive is the lowercase. So that's the short. So it's going to be 2 little t's. Homozygous, same, recessive, lowercase. So 2 little t's. So these are our 2 parents. You can see each parent has 2 letters because they have 2 alleles, one from their mom, one from their dad. And now we have 2 parents. They're going to come together and they're going to make some children. So what we're going to do is we're going to use our Punnett square here. We're going to take our parents' genes And we're going to take what will be their gametes and place them on the outside of the Punnett square. You think of putting sperm on one side, eggs on the other, and they're going to mix in the middle to make children. It doesn't matter if you put one on the top or one on the side, where you put them, as long as you put one parent up here and one parent on the side. It's important when we go to split up the gametes. We're going to split up each one from each parent. So we'll put a capital T, say here, the other capital T, say here, a lowercase t here, and a lowercase t here. Now I could, like I said, we make 4 sperm and 4 eggs every time we go through meiosis. So I could do a 4x4 box, but remember, 2 of those sperm or 2 of those eggs are going to be the same every time. That's what lets me use the 2x2 box. 50% of the sperm is this, 50% of the sperm is this. 50% of the eggs are going to be this, 50% of the eggs are going to be that. So we'll see how this works right now. We're going to put one parent, the little t on the side, and one parent, the big T on the top. And I'm just going to separate them into the different columns and the different rows. And notice that, let's say this is dad for whatever reason. He's entirely up here. Mom is off to the side here, right? So one parent goes on the top, one parent goes on the side. Now what we're going to do is we're just going to fill this Punnett square in. And the way we're going to do it is we're going to do it by carrying the letters down and across. So think about this as a column, and that column has all big T's from, let's say, dad. So a big T here, a big T here. Same thing over here, a big T's coming down, big T, big T. So we're going to go ahead and just carry those down. Those big T's are coming down, 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 down. Same thing over here, we're going to bring the little T's across. So that little T will come across to fill this box, come across to fill this box. This little T will come across to fill this box and this box. So we're going to carry those in from the side. So all we're doing is mixing and matching. You think of this box, big T, little t. This box, big T, little t. It's just like matching the top with the side. Big T, little t, big T, little t. In this case, my entire box is big T, little t's. But this won't always be the case, depending on your problem. So now once we figured out and we filled out our Punnett square, it's time to actually see, well, what do I have for children here? What do my children come out looking like? So the first thing we're going to do is we're going to figure out what we call the genotypic ratio. So remember, genotypes are the specific allele combinations that we can be. Remember, there's always 3 of them. The first one is homozygous dominant. Well, how many of my children are homozygous dominant? Homozygous dominant would be big T, big T. Well, that's not big T, big T. Nope, nope, nope. 0%. None of these boxes are big T, big T. The second genotype that we can be is heterozygous. Big T, little t. Well, that box is big T, little t. That box, all the boxes are. So if every box is 25% of my children, or 25%, that means 100%. There's a 100% chance that a child from this cross could be big T, little t. And then you can think about homozygous recessive, little t, little t. Nope, 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 nope. None of the children in the box are little t, little t. So 0%. None of the children will ever become little t, little t. So this cross is pretty cool in that 100% of the children are going to be heterozygous. I know for a fact that all of my children are going to be heterozygous. And then the last step is just to figure out, well, what those children actually look like. So remember, we always have 2 phenotypes, the dominant and the recessive. So the first one is tall. Remember, that was the dominant, big T. Well, is this child going to be tall? Well, actually, yes, they are, because they have a big T, and dominant traits always win. So this child in here, who's heterozygous, is going to be tall. And so will this one, and this one, and this one. All of these boxes represent tall children because they all have a capital T in them. Dominant always wins. So 100% of my children will be tall. How many are going to be the other phenotype? Short? None of them. Because the only way you can be short is with a little t, little t. 0% of the children have little t, little t. So that's how you go ahead and solve these problems and kind of predict the traits of your offspring. I'm going to have you practice a few of those right now.